Today is the November 3rd meeting of the working group. We have a single speaker from Carnegie Mellon, who's going to be talking to us about somatic evolution in cancer cell lineages. As always, I need to remind you that the meeting is live streamed, recorded, and archived for later viewing. And we hope that people who are not able to attend right now will be able to watch it later. As always, Tom and I are available to take your suggestions. Lorenzo and Joel are also available, and Jim Sluka as well. Please let us know what we can do to make things work better. We have various social media communication channels. It would be helpful if people can use those for us. In particular, there is the IMAG MSM Wiki page, which has an archive of slides and talks. And there's the YouTube channel that has all of the recordings of the seminars. There's also a LinkedIn page and a Twitter feed, MSM Viral. People use that Twitter. That can help us perhaps improve attendance for these talks, which are always very good and deserve to have bigger audiences. It was the second anniversary of the working group. We have delayed the celebration to December 8th. It's going to be the December 8th meeting. Please help us build the presentation. There's a slide deck that's being put together. We would love to hear about your experiences with the working group. We realize that many people have developed new collaborations, have written articles, have had grant funding that grew out of the working group, and we know about some of that. We'd love to hear more about your experiences, also about history and about ideas for what we can do going forward. So please do help us make that an interesting discussion for December 8th. Are there any announcements in the room today? Reinhardt is, as usual, leading the Digital Twin subgroup. It's going to meet tomorrow. It meets alternate Fridays. If you want to join it and you're not already on their mailing list, please do email Reinhardt and get put onto the list. The immunology subgroup is meeting regularly again. That's coordinated by Veronica Zarnitsina. If you would like to join that, please email her. And if you can't get the email addresses from the slides, I can put those in the chat for you if you ask. Are there any other announcements before we get started? Coming up November 10th, we have two speakers from Physique who are going to be talking about their work. November 17th, we have a second visit from Nova Discovery. Nova Discovery has been building some very interesting computational platforms for doing virtual clinical trials. They have proprietary virtual clinical trials, but they also are developing open source software for creating virtual clinical trial models. So we're going to be hearing from them again. Then December 1st, we have Baylor Fain from Texas Christian University speaking to us. And then we have our second year anniversary presentation and discussion on December 8th. Please do help us recruit speakers. Suggestions would be welcome. There's an efficient Google form that you can use to provide your ideas, and we'd appreciate that. So I don't want to take more time from our speaker. Today, we're very pleased that Russell Schwartz from Carnegie Mellon is going to be talking to us about modeling and inferring somatic evolution in cell lineages. That's a topic that I know is of interest to many people. It doesn't perhaps sound directly like viral pandemics, but the group here has a lot of interest in cancer and cancer immunology.
It's really a pleasure to be able to speak to this group. I realize that this is probably not exactly the typical topic for your group, but I think it's an interesting one. I hope there will be some useful lessons for those working on multi-scale modeling in other contexts. So hopefully there will be something to learn for the people here. So I want to talk about a problem that my lab has been working on, talk about it from a few different directions. And this is a particular problem of modeling and inferring somatic cell evolution in cell lineages. And this is a problem that is originally largely motivated by studies of cancer progression, although it's now appreciated that somatic evolution is much more common and much more important than just the case of cancer progression. But although I will be primarily focusing on what we're learning about cancer. And all the work I'll be talking about is essentially flowing from the idea of cancer as a disease of evolution. And what I mean by that is that it's been understood for about 50 years at this point that cancer is basically a disease governed by evolutionary processes of diversification and drift and selection. Essentially, we would generally believe that cancers begin with a single initially healthy cell, and that healthy cell undergoes some kind of mutation that sets it on the path to tumor development. We'd argue that in many cases, one of the first mutations, if not the first mutation, is something that is going to be producing some sort of somatic hypermutability, so a propensity to generate many more mutations over successive generations. So typically over successive generations of cell division following that initial mutation event, we get a proliferation of genetic heterogeneity, and that heterogeneity creates opportunities for selection. So different cells may just by chance have selective advantages that allow them to proliferate more rapidly, escape various controls on cell growth, and so forth. So this has been understood as a principle for thinking about how cancer develops for, as I say, about 50 years at this point. But it's relatively more recently that's moved from being a way of thinking about cancer to really a source of very practical tools for making sense of cancer genomic data. And that's largely been through a class of methods known as tumor phylogenetics, which is basically the idea that if cancers are evolutionary systems, then we can bring computational methods for understanding evolutionary systems to understanding genetic variation in cancers, and in particular phylogenetic methods, so evolutionary tree building. This idea has taken on a number of different forms. And I want to focus in particular on the problem of inferring individual cell lineages in cancers. So looking at single tumors and understanding what the evolutionary history of the cells in a single tumor are. This is an area that my lab had gotten into a pretty long time ago at this point, starting with the question of just how would we infer a tumor's evolutionary history? How could we look at a tumor and figure out what the evolutionary history of the cells in that tumor were? This is something that we began really before it was possible to do tumor sequencing. At the point where we had gotten into this, the first human genome sequence was relatively fresh, and it wasn't possible to do human genome sequences or tumor sequences on anywhere near the scale we would want to look at evolutions of different tumors and be able to compare across tumor populations. So the field really started with various kinds of pre-genomic technologies. And in particular, we started this area of looking at evolution of single cells using fluorescence in situ hybridization, or FISH, which basically just means attaching fluorescent probes to single cells in cancers and using these probes as markers of evolution. So in this example, a very early data set, basically a more than 20-year-old data set at this point, we have 2 probes attached to each cell from a population of single cells in a tumor. And even just those 2 probes give us a way of separating distinct cell populations, constructing essentially histograms of copy numbers of these probes in single tumors, which turns out to be a useful thing to do for tumor evolution, because cancers to a large degree evolve by by gain or loss of genetic material. So amplifying oncogenes that give selective advantages to tumors, or losing copies of tumor suppressor genes that remove controls on cell growth. And so we were able to build these histograms and use phylogenetic methods to describe how a histogram like this could have been produced by an evolutionary process. And then by collecting these across many different tumors, build consensus models of evolution of the tumors, such as the one shown here, showing a consensus from this 2-probe data set for about 100 different breast cancers. And even with these very primitive data sets, we could start to see something about the pathways by which cell lineages evolve, how those differ between different cancers, and sometimes even between different cell lineages in a single cancer. Nonetheless, that was quite a primitive kind of technology relative to what we would mostly work with today, although these FISH technologies have continued to develop and I would argue continue to be useful for these kinds of studies. This is an example using a more recent data set and more recent algorithms for studying them, in which in this case we're using a version of multiplex FISH, which allows you to get larger numbers of probes, in this case 8 probes per tumor. In practice, you can really get up to a few dozen, maybe as many as 50 or so probes per tumor. And as the technologies have gotten better, they've gotten better at being able to distinguish different clonal populations and be able to distinguish some important events in tumor evolution, like a genome doubling that's represented in this more recent phylogeny. And they remain quite useful for studying some of these kinds of events in tumor evolution. Nonetheless, the field has changed quite a bit, and largely it's been rewritten by sequencing technologies, like so many other things in studies of biology and especially evolutionary biology of different kinds. So much of the field was really rewritten as it became possible to start doing sequencing on large scales, and in particular to be able to start sequencing large cohorts of cancer patients or large cohorts of tumors. In the earliest versions of this, it still wasn't possible to get down to the single cell level. And so there were some hard trade-offs between these earlier pre-genomic technologies and the sequence technologies. Sequencing at the time could basically sequence a whole tumor or a large region of a tumor, mixing sequence from large numbers of different cells that would be present within the heterogeneity of that tumor. And so different classes of methods needed to be developed that could work with this kind of mixed genomic data. Essentially what are now known as genomic deconvolution methods, where we could take data on mixed tumor samples, essentially kind of matrices of data representing some kind of genomic markers across different sets of genes or genomic regions, different sets of samples, either from a same tumor, different tumor sites, different regions of a tumor, in some cases different tumors, and computationally deconvolve these to identify major cell populations and how those cell populations are mixed in different tumors. So a deconvolution problem where we could then infer what different tumors look like and come up with a different kind of evolutionary tree, where we could then get away from some of the limits of the FISH technology, be able to look in an unbiased way at the entire genome, but also be able to do this in a way that we could discover novel gene variations, discover novel genes that we may not have realized were important to our tumor evolution, and come up with a tree much like the one shown here, where we could get maybe a fairly limited resolution of different cell types, identify maybe a few common cell types, which themselves are really conflations of many genetically distinct cells, but have the advantage of being able to look at them in a whole genome sense. So this really gave us some significant trade-offs with the FISH technology. And it's important to emphasize here that sequence had big advantages over FISH, but it did not displace FISH and the earlier pre-genomic technologies. There were still trade-offs between them, in particular that the FISH technologies primarily could give you much better resolution for different cells, so true single cell resolution, at the trade-off of giving you very few markers, while the sequencing technologies could give you whole genome marker sets with the trade-off of fairly limited resolution for single cells. Now, in theory, this limitation was removed by the advent of single-cell sequencing, and really work of Navin et al. That brought single-cell sequencing to the cancer space, showing that it was possible to sequence single tumor cells or nuclei of single cells and produce data sets where, in theory, you could get all the advantages of whole genome analysis and all the advantages of single-cell resolution. Now, I say in theory because things weren't quite that good in practice and still aren't quite that good in practice. In part, this is because of some technical limitations and artifacts of single-cell sequencing. So depending on the technique you use, you can have large error rates, large amounts of dropout, although these are manageable problems, but more so because there are significant resource limitations. Single-cell sequencing is a fantastic technology, but it is an expensive technology, and it's still not really possible to do this at the scales you would need to do large numbers of single cells in large cohorts of cancer patients. This is something you could do with FISH. This is something you can do with bulk sequencing. It's not yet something, even today, a decade after this first single-cell sequencing work, not really something you can do on those scales for bulk sequences yet without a very large investment of resources. And what I'm getting at here is something I think has been shaping a lot of the work in this field, and I hope it can be seen as a model of a lot of things going on in biology today. And that is that there really isn't one dominant technology. There are a variety of different ways of looking at this problem with different technologies, and each has its strengths and weaknesses and trade-offs among them. And so a lot of what's been happening in this cancer field, a lot of what my lab is doing and what other labs are doing in this space, is really more and more turning into a multiomics problem. So a problem of saying that we have different data sources, none is completely dominant over the others, each has its advantages and disadvantages, and where we can really do the best work is in trying to figure out how to use combinations of data more effectively. So this is an example of a technique we had put together by talking about some of these trade-offs between bulk and single-cell sequencing and asking really, if we had a finite amount of resources, how would you best want to invest them in some combination of bulk and single-cell sequencing? And what we essentially proposed was that really what you want is something close to single-cell resolution and large cohorts if you want adequately powered studies. You're not going to get that from just bulk sequencing, and you're not going to get it from just single-cell sequencing. And maybe you can do better with a combination of the two. So what we had proposed was basically a kind of optimization problem where you can mix combinations of bulk and single-cell sequence, really with the goal of having bulk sequence on a fairly large number of tumors and limited amounts of single cell on those tumors, and pose the simultaneous problem of deconvolving the bulk data, doing that in a way that's consistent with the single cell data, and doing it so that both your observed and your inferred single cells lead to a phylogeny that is evolutionarily plausible, essentially something like a minimum evolutionary distance phylogeny. Pose that as a single computational optimization, and then simultaneously solve the problems of deconvolving your data, explaining how different cell populations are mixed in different regions of a tumor and how they go together into a phylogeny. And essentially the conclusion of this work was that you can indeed do better with these two technologies together than you can with either one alone. And you also do better by solving all of these problems simultaneously than by solving any of the problems in isolation. The solution to each of them helps you solve the others. And so it turns out to be productive to solve them together, even though in some sense it's a harder computational problem. And that's a lesson that we found repeated over and over as we reasoned about what we can do with different kinds of technologies. So I mentioned that the FISH technologies were still not obsolete. These still have an important place. And this is something we examined by combining single-cell sequence and FISH. In principle, single-cell sequence is just unequivocally better than FISH. You get whole genomes at the single-cell level instead of a few markers at the single-cell level. In practice, FISH has a very big advantage in that it's more scalable. You get much larger numbers of cells. You get better quantification. And it also has a subtle but important technical advantage in giving you absolute rather than relative copy numbers, which turns out to be very useful for analyzing ploidy change events, so changes in chromosome number in cancers. And so we were able to pose a problem of bringing together fish and single cell sequence data through a kind of joint clustering problem, where we imagine there's a latent data set that contains our full single cell sequence data. And we're seeing it through two kinds of incomplete windows with these two different assays. And we try to find the clustering of data that is maximally consistent with these two assays. And then that gives us a set of inferred sequences with absolute copy number that we can use, in this case, to do a much better job of understanding, again, how genome duplication events play out in cancers, and rewrite some of the common understanding, we believe, of how these events occur in tumor evolution. In particular, seeing it as more of an ongoing process based on these data, rather than an early event in tumor evolution, as was commonly assumed before this. And if you can do better with single cell sequence and fish and with both single cell sequence and bulk sequence, why not bring all three together? And we tried that as well, and we could show that all three together does better than any one or any two together. Essentially, we can pose the problem of bringing all of these together, deconvolving the bulk data, matching inferences of clonal frequency between the two single cell technologies, getting a parsimonious evolutionary tree, and matching basically the common probes between fish and single cell sequence. We do better by bringing in all these pieces of the optimization. And we also, again, do better by solving several problems at once. The problem of inferring what single cell clones we have available, inferring how these are distributed among different regions of a tumor, and inferring the evolutionary trees that explain those single cell sequences. So that, again, is something that we find is supporting this idea that really multiomic inference is where we really need to be going to be doing the best we can from these different data sets. And that question of bringing in these different technologies is just one axis on which it ends up being useful to try to think more broadly and effectively solve broader classes of problems together if we want to get the best answers to each problem individually. Another dimension in which this occurs in the cancer case that is maybe especially important in cancers, but also so in other evolutionary problems, is in bringing different classes of genetic variation together. Much of the work in tumor phylogenetics is focused on point mutations. In a sense, they're the easiest thing to study computationally. A few labs, including my own, have put more attention into copy number changes, which is a harder computational problem, but we'd argue a more important one for functional adaptation. But there's been very little on broader classes of variations that more or less fall into what's called structural variations. So things like large-scale duplications, rearrangements, inversions, translocations. And this is another thing we've worked on, trying to bring into these phylogenetic inferences. First, bringing these together with just copy number changes, and in the most recent work in this space, trying to bring together the structural variants, the copy number changes, and the point mutations all into a single evolutionary inference. And again, what we find from this work is the same lesson, that even though in some ways it's a much harder problem to try to solve all of these at once, you actually do better than you would looking at any of these in isolation, because they're complementary to one another. And each of these kinds of data has its own, or variants has its own advantages that helps you explain the other variant type. So we actually do better solving all three at once, building phylogenies that integrate all of these kinds of events, and again, inferring the evolutionary trees of the clones we infer and how these are distributed among different tumor regions or tumor sites. So what I'm describing is some work from my own lab, but this is a lesson that has been repeated from many labs that are now working in this area, that there are numerous tumor phylogeny methods available for different kinds of sequence data, different kinds of variations. This is a small fraction of what is now available for many different groups working on this area. I would guess what I'm showing here is now maybe 10 to 20% of the tumor phylogeny methods that are actually out there. And that's just talking about DNA. There are whole other domains of this problem when we start bringing in other omic technologies. So this is an example of some methods we've been developing, studying the tumor evolution problem from the point of view of RNA. And that is another dimension in which we see very different kinds of pieces of the puzzle of tumor progression, with DNA, you're seeing what's really happening in terms of the evolution of the genome of the tumor cells. RNA shows you a little of that, but gives you a lot of other information about what is functionally happening in terms of adaptation of expression modules, so things that are telling you something about what is functionally changing in the tumor cells as they're developing. But more than that, it's telling you about all the non-tumor cells, because a cancer is not just a mass of tumor cells, it is a collection of tumor cells, non-tumor stromal cells, various kinds of infiltrating cell types, immune cells, obviously quite relevant to what I know some of you are working on. And the tumor is really the interaction of all of these different cell types. And RNA gives you another window into what cell types are present, how these are changing across the progression of the cancer, and how they're influencing one another's behavior in terms of different pathways that are being activated or deactivated as these cell types interact. And that is yet another space where multiomic technologies are becoming more important. This is just an illustration of a method we put together for combining bulk and single cell RNA-seq, which is yet another problem that turns out to have some interesting applications in cancer development, and to be able to reveal things that you sometimes can't get from one technology or the other, or can't get in situations where you just don't have access to all the genomic technology you'd like to have available. And what all of this is really getting at is that cancer is a space in which there are an enormous combination of methods we could develop. Pretty much anywhere in the space of different biotechnologies that are available, different kinds of evolutionary markers, different kinds of variant types. You can pick a point in this space, and I'd argue that you could develop a tumor phylogeny method, and you're going to learn something from that method you wouldn't learn at any other point in this space. And that's true from every point in this space, and it's true for every combination of points in this space you can think of. There's just a vast space of methods we can develop, and what that is really getting at is that cancer, and I would argue cancer is really in some ways the leading edge of systems biology in general. This is a space where more and more trying to get at the details of the biology is a problem of dealing with very complicated biology, very complicated heterogeneous data, and trying to figure out how to make the most of that. And all of that is a very long introduction to the question of what we want to do next. Why are we doing this? What are we actually learning from it? And how can we optimize our ability to learn from this in the future? And one of the first things I think we learned from tumor phylogenetics that I think is not always obvious, and in some ways has to be almost relearned by everyone joining the field, is that you don't learn anything from building one tumor phylogeny. Most of what's happening in a tumor phylogeny is just stochastic. It's purely random chance, and you can't tell what's random chance from looking at one tumor. It's really only when you start looking at groups of tumors that you can start to figure out what is actually consistent, what is actually meaningful. And when I say meaningful, what has predictable, reproducible information that can tell us something about the future of that tumor. This is a piece of the talk that when I first wrote the abstract, I hope to spend a bit more time on. I realized I wouldn't have time to give more than a vignette about it, but I really want to get to the main conclusion here, which is that the past of a tumor tells us quite a lot about the future of that tumor, but only when we have adequately powered studies and sufficient data that we can extract what is meaningfully predictive from these phylogenies. So if we just look at a phylogeny, there's no way to tell what edges, what nodes in that phylogeny are meaningful, but we can extract features from these, quantitative measures of the evolutionary history, things like what evolutionary preferences are there for gain or loss of particular kinds of mutations, gain or loss of broad classes of mutations, mutations in particular genes, preferences for building broad or narrow phylogenies, shallow or deep ones that tell us something about the overall evolutionary process. And that kind of information, when brought into a statistical or machine learning paradigm, turns out to have useful predictive information. This is just an early illustration of that from some of our very old studies, showing that various kinds of predictors, of features we can extract from, in this case, cervical cancer phylogenies, give us significant predictive power to distinguish progressing, that is metastasizing cancers from non-metastasizing, whereas the raw data without the phylogenies on which those were based basically do no better than chance at that task. And that's a lesson we've observed over and over for different kinds of tumor phylogenies, different data sources, different kinds of features we can extract. This is another version of this from a kind of phylogeny using bulk genomic data to mix, to basically do a mixture of kind of DNA and RNA data to represent, to kind of extract different overall evolutionary pathways in the tumors and show that you can get significant separation and survival prediction. And this is a newer version using some more advanced kinds of machine learning to show that you can bring together different kinds of information about a tumor, more classical clinical information, information on driver mutations and various phylogenetic features. And each of these classes of mutation individually as well as in combination gives you additional power to separate tumor types. And for various tumors, various kinds of progression outcomes, various kinds of sequencing data, reach the same conclusion that each of these pieces of information, when we have sufficiently powered studies, is giving us the ability to make useful predictions about the future of those tumors, in this case, predict various kinds of survival outcomes. So the overall conclusion here, and I know I'm skipping over really the details of all of this statistics and machine learning, but the overall conclusion is on this slide, that there are many ways of building tumor phylogenies, many ways of taking different kinds of data and putting these together to extract different kinds of markers. And pretty much no matter how we do this, we find that there is variation in what we refer to as mutational phenotypes, variation in how the tumor generates mutability and how it's evolving. Some tumors have high preferences for particular kinds of mutations, some have high or low mutation rates, some have high rates of structural variation and low rates, some have high rates of copy number variation, some have low rates. And no matter how no matter what tumors pretty much we study or how, we find this variation. And pretty much no matter what paradigm we look at, we find that it has predictive power for future progression of the tumor. Obviously, it matters how we ask this question, how much predictive power we can get. But if we bring together the right kinds of data, the more we learn about these tumors, the better we can build these phylogenies, and the more accurate they are, the more actionable information we get about the future of these tumors. And this is a conclusion about cancers, but I would argue that cancers have often been the leading edge for genomic technologies and computational technologies based on them. I think this is a conclusion that is going to turn out to apply to a lot of other problems in systems biology. And that really brings me to the last part of the talk, the last thing I wanted to discuss here, which is really kind of work in progress and really getting into really the modeling and simulation part of this talk. So what do we do with this information as we know that we're moving into this space where progress really is increasingly depending on very complicated multiomic study designs and how we can make the most of these? And I think more and more this is a question we need to deal with in a more rigorous way in the field. How do we design an informative study in this space where we know that we are dealing with these complicated multiomic problems? Because there are now many different options available to us for study in this area. Many ways we can take apart a tumor, many ways we can use different kinds of time series data, generate different kinds of samples, different kinds of omics data, apply different kinds of computational analyses to them. And what all of this is giving rise to is the fact that there is a complicated space of study designs you can consider and difficult trade-offs within this space. If you are trying to study whether there are mutation rate variations that have predictive power for the future progression of cancers, you have to ask the question, how much of our investment should we put into sequencing more tumors versus more sites per tumor, or more time points per tumor, or more single cells per tumor? How do we trade off the use of whole genome versus whole exome versus targeting sequencing? How do we trade off how much bulk sequence versus how much single cell sequence to do, how much liquid biopsy to do versus how much profiling of our solid tumors? And that's not even getting into all the questions about what algorithms algorithms we apply to these, what approaches we're using to analyze the data. Basically, we have this huge space of methods, and what I would argue is that this is a space where we need to be thinking much more carefully about how we're designing our studies, because vast amounts of resources are being put into these, and really it's mostly being done with hand waving at this point. So mostly being done by people pretty much kind of saying, we want different technologies, we think they'll be informative, maybe doing some power analysis to say, okay, this is the study we're proposing, we can show it's probably powered for this particular question, but not really going to the question of what would be the optimal design, what would be the optimal use of resources for that question. So we've been developing, and we put out a first version of a simulator to try to give us a basis for analyzing these problems, because I think at the moment we just don't really know enough about this problem to have a firm analytical handle to be able to do any real analytical modeling that can tell us how to design a study. For the moment, I think this is really a question for simulation. Can we simulate the evolution of tumors, simulate many different trajectories, simulate the outcomes of experimental decisions applied to these tumors? How do you sequence your tumor? How do you break it up? What regions do you look at? What time points? Simulate the sequencing technologies or other genomic technologies applied to those tumors, and put all of that into generation of artificial data that we can then plug into optimization schemes to try to design our studies. I know I don't have a lot of time left. I didn't plan to say much about how we're generating the tumor sequencing, because really it's a fairly standard kind of population genetics model, a coalescent model for generating genetic variation in a tumor model, extended in various ways for things that are important to tumor biology, like the different kinds of genetic variations we see, how these might be distributed spatially or to different sites within an infected individual, as well as some of the special kinds of technologies that are important in tumors, like for example, liquid biopsy that's becoming important. Add on top of that simulations of the biotechnology, so simulations of what it would look like if we are drawing a sample of single cells and we're drawing bulk sequence, and we're doing this from different regions within the same tumor, and then put that into a simulator model where we can generate lots of simulation trajectories and start to ask questions about study design. So in a very simple version of this, we can do something more like the standard practice today, where we can propose a particular study design, simulate data under that design, and ask if we would be powered to answer a particular question, like if we would be powered to detect a particular variation in mutation rate between two different subclasses of tumors or different cell lineages within single tumors, and be able to use simulations to ask a question like that. Are we adequately powered? Can we show that? But really where I think this is going, and where I say this is work in progress because we really haven't solved this yet, is the question of how we would optimize that design. We can do some parameter scanning. We can start to say, okay, maybe we haven't fixed our design. Let's try scanning over different choices, like the amount of depth of sequencing we want to apply, and see how that affects the study design. But where this really needs to go, I think, is solving the general study design problem. To be able to describe a loss function saying, okay, what is it we're trying to do with this study? What do we want to be powered to do? What would the optimal outcome or a better outcome look like here? Maybe specified in terms of sensitivity for different kinds of things we're trying to observe. What are our constraints? So constraints might be resources. We have a finite amount of money we can apply, let's say a finite number of patients we can recruit, and pose this as a constrained optimization problem, and really come up with an optimal study design. We've done basically some toy versions of this. This is an example allowing us to vary a few different parameters to pose the question of how can we maximize our power for identifying structural variants and point mutations in a study design where we're able to vary a few different parameters of our sequencing, and pretty much brute force the problem of being able to consider how this varies across different parameters singly and in combination, and come up with an optimized study design. But this is something I'm really putting out there as something I think is an unsolved problem for the moment that we're working on, that others may want to work on. And even those who aren't working in cancer, I think cancer is a space where I think there's going to be a lot more work in this area than there is now. I think there's a need for it. And as I say, cancer is sometimes a leading edge for other problems. Some areas are ahead of the cancer field in this way, some are behind. I think this is something that really anyone working on multiomics should be thinking about solving more. And there's a very important role for simulation as well as optimization here. So just to kind of wrap up and draw some conclusions, somatic evolution is a problem that I think is like many problems in modern biology, in that the more we get into it, the more we're digging into very complicated biology, complicated data, and complicated computational models needed to make sense of it. And progress really depends on computational methods that can manage that complexity and deal with some of the heterogeneity of biotechnology, biology, and computation needed to study this. Simulation in particular, I think, is emerging in this space as a crucial part of the computational toolkit. There are other aspects of cancer where it's already quite important, but I think not so much yet for the study design multiomic space. And it's really, I think, going to be a growing area alongside traditional algorithmics, growing use of data science and machine learning, and various other kinds of paradigms, both experimental and computational, managing this complexity. We're working on many other areas, improving the multiomic optimization methods, the machine learning, and working on this problem of simulation and study design. But I'll just close by thanking many who contributed to this work, members of my own lab, collaborators within Carnegie Mellon, collaborators outside of our institution, and many, well, those who provided funding for different aspects of this work, since I'm really describing work over a period of quite a few years, and those who provided data, because we're quite dependent on some of the large cohorts like the Cancer Genome Atlas, the International Cancer Genome Consortium. And of course, thank you to all of you watching for your attention. I hope this has given at least some useful perspective, even for those of you who don't work on cancer biology. So thank you.
Okay, thank you very much. Time for some questions.
I have one. Do you see any usability of all the findings and all the collections you've been doing on this predictability? Do you think an agent-based model could help in any sense for you to advance more discoveries?
Yes, an agent-based modeling is used quite extensively in other questions in cancer studies. So a lot of what's happening in tumor evolution, we're looking at this primarily from the phylogenetic perspective here, so reconstructing from data, but there's a lot of work looking more from the bottom up. So if you make certain assumptions about the tumor, how is it actually going to evolve? And our coalescent models are kind of a simple version of that, but really a lot of the interesting work in this space right now is using agent-based modeling to ask more comprehensively, how do different clones in the tumor interact? How do they interact with the microenvironment? How do they interact with the infiltrating immune cells and other factors in the body? So definitely there's a big place for agent-based modeling and in improving simulations and for this question, as well as many other questions in tumor evolution.
Other comments and questions? I think that Joel's point was a useful one, because what we have some data scientists, I think mechanistic modeling tends to be the home for many of the people in this audience. And so trying to think about how to interface those kinds of bottom-up mechanistic models with the kind of data that you generate. So you made the point that you can't really do much with the phylogeny from a single tumor. You have to look at populations to get patterns of evolution. And this is a problem with simulation as well. You have stochastic simulations, and they'll give you, each one will give you some trajectory. In addition, even the biggest agent-based simulation has 10,000th or a millionth as many cells in it as an actual tumor would. And so there are issues about how do you simulate in a mechanistic way, how do you simulate evolution? Because you have to at least, if nothing else, turn up the probability of mutations enormously compared to the actual one to be able to see anything in a simulation. And so I wondered if you had wanted to speak to that, sort of how do you have, what do you see as the opportunities to interface these two very different and important points of view?
Yes, and it's actually a problem we're struggling with in my lab right now. So the coalescence simulations I talked about, this is, it's often referred to as backward simulation, basically going backward in time for efficiency reasons, because it lets you simulate very large populations, but it has to make some hard assumptions. So one of the things it has a lot of trouble dealing with is selection, and getting selection in the coalescent model is a hard problem, but of course it's quite important in cancers. And this is a tension in our work. It's really, I think, a tension in the field that we're trying to simulate these processes that involve vast numbers of cells and often very long time. So a tumor may be developing for a decade or more. And backward simulation has big advantages there, but there are big trade-offs that forward simulation, agent-based and other mechanistic models can give you a much more realistic simulation of what's happening, and drop all of these assumptions are really quite problematic in the coalescent. So I wish I could say I knew the right fusion of these two technologies to really get the advantages of both, which is really what we want for the problem we're trying to solve here. So that's the problem we want to solve. I don't know the answer, but it's a very important question.
Maybe following up, you had a nice slide where you talked about sort of identifications of clusters of cell types, and whether you really saw a clustering of in this case genotypic rather than phenotypic behaviors, and how reproducible those were from tumor to tumor or time to time. I mean, there are two questions there to me always. One is I mean, ultimately selection happens at the level of behaviors. It happens at this level of phenotype, not at the level of genotype. And things like motility, invasiveness, there are many different genetic ways you can get the same change in behavior. And so you don't necessarily expect to see a single genetic signature for increased in metastatic potential, for example. P53 is a little bit different. It seems to really be a choke point, but in a lot of these other things, you see this very dispersed set of possible ways of doing the same thing. And I wondered, with all the experience that you have, how much do you see real convergent evolution? In other words, the strategy for the cells tends to be the same genetically, and how much do you see many different approaches to solving the same problem, which is how do I move in the tissue tumor? How do I avoid stop signals? How do I do immune avoidance and so on? With the extensive studies you've done, what's your instinct about how this mapping works?
Yeah, that's a great question. Honestly, this relates to a question I think about as an evolutionary biologist, that there's a classic evolutionary question about the reproducibility of evolution. If you could roll time back to the beginning and just run it over and over again, would you get the same organisms again and again? And cancer is really a space where that is actually a natural experiment that has been done. You more or less have the same selective landscape that has been navigated by billions of tumors over the course of history of species. So we can start to get at that question. And the answer, as with most things in biology, is it's complicated. There are definitely recurring themes that come up over and over. Every tumor at some level does its own unique thing. It will have its own unique pattern of mutations. but there are absolutely recurring themes. A lot of this is captured in this very famous review by Hanahan and Weinberg on the hallmarks of cancer, which really captured this idea that there are a set of functional effects that come up over and over. There are different ways they can occur, but not an infinite number of different ways. And you'll see over and over again tumors navigating the same problems through a finite number of more or less similar pathways. And I think that's consistent with what we see, that there are subtypes of tumors. They mostly will fall into small numbers of categories where they're mostly sort of doing the same thing, but each one is going to be doing something unique. They're going to have their own unique set of mutations. Sometimes some high impact mutations will occur over and over, but then they'll be in a background of other mutations. They're probably adding more minor effects. So I don't know if that's actually answering the question, but again, it's a great question, and I think cancer has a lot to say about it.
Right. Well, I mean, that's precisely where I'm coming from, in the sense that the hallmarks of cancer are phenotypic hallmarks. They're behavioral hallmarks. They're not genomic hallmarks. And so the question, I guess, is right? I mean, sort of classically, you look, right, P53 is knocked out at most solid tumor, maybe at least half. But there are a couple of other APC, a few other genes that tend to get knocked out pretty commonly. But then you get this background level of this gene is mutated 1%, 0.5%, even within cancers that are the same kind of cancer, in quotation marks. And so there are these clusters of different ways you could solve those functional problems. And it seems to me that the data sets that you're generating, especially the ones where you were talking about the RNA and linking to functional terminology, allow you to perhaps to begin to answer the question of what's sort of the cloud of mutation space that allows you to solve those functional problems.
Yeah, and I'd say that kind of principle, we've used it for trying to improve the phylogenies, or in the RNA case, it's not strictly speaking a phylogeny, or these models of populations of cell types. And you do get a lot of power of thinking about things that way. So thinking of your evolutionary markers as a change in a pathway rather than changes in individual genes, just collecting the data that way cleans it up a lot. It gives you more power for these evolutionary studies. And in the process, it's revealing something about what's happening at that more functional level. And it does help you collect the tumors better. So identify the tumors that might not have the same genes mutating or changing expression, are in some sense altering the same pathways. So, again, I don't have the answers, but I think you're absolutely right. There's a lot to learn from these data about what's going on there. It's definitely something we're seeing in the data. I don't think we've really fully exploited that yet, though.
Other questions?
I have another one. Your technique is very focused on cancer, as you were saying, because it's your main research. And you also mentioned that this could be applied to other things, right? And I was just thinking about the definition of heterogeneity in the tissue, right? Sometimes when we are modeling something, we want to know all the cells that are there and what they're doing, how important they are to be represented. Do you think some of your techniques can help us to give our answers? Because most, I want to speak for all of us, but for example, I'm not a biologist, so having a way to shortcut and understand exactly how some of those genes are connected to some behaviors and what are the things that they use to generate those behaviors can be very helpful. Do you think there is a space in your methods to do some kind of data application?
Yeah, I'd say absolutely. It's getting farther from my own expertise, but there's a lot in common between tumor evolution and viral evolution. And you often see some of the same things that make tumor evolution tricky. You also see in viral evolution, for example, often very elevated mutation rates. And the similar kinds of questions come up. So the virus needs to escape a vaccine, or it needs to escape some aspect of the immune system, or jump to a new host. It's got a selective landscape to navigate and maybe different pathways to pursue there. So again, it's really getting beyond my expertise, because I haven't worked much on viral evolution, but I think the same high-level questions and some of the same kinds of computational ideas are likely to apply.
Okay. Time for a last question, if anyone has one. Reinhardt, you sit in a cancer-focused institution.
Well, so like most medical schools, we have a cancer center. I used to work in cancer, but not anymore. What interested me in your talk, which was a great talk and is really interesting. I just recently sat on a cancer systems biology study section. So a lot of the questions that you raised were discussed there. It's once you have the data you collect and you have models, whether they're Boolean networks or agent-based models or other kinds of models, what are some of the questions that you would hope one would be able to answer?
It's a very broad question, is that there are a lot of things you want to answer with cancer. And sometimes, like with those evolutionary questions, cancer can be really a window into other kinds of basic research, a window into evolutionary biology, into systems biology more generally. But of course, it's also a major killer. And a lot of the questions really trying to get at is, what can we learn about tumor evolution that will be actionable for the clinic? How can we improve personalized medicines? And what can we learn about the evolution of one person's tumor that is predictive about whether it will respond to a particular drug, whether it is ripe for immunotherapy, whether the patient has a good or bad outcome, whether there's something that can help you stratify patients for clinical trials to discover that maybe a whole class of drugs might be benefiting a subclass of patients you didn't know before. So those are really a lot of the questions we're trying to focus on. What is actionable for either the clinician, what is actionable for drug discovery, and trying to get at those things. But really, cancer is a window into so many different processes. There's almost no end of questions you could be asking.
Is there a particular type of data you think would be very helpful that's not being collected at the moment?
I'd say it's not necessarily a particular kind of data. I'd say it really comes down to, for us, a lot of the multiomic data. What we're really starting to understand is that it's not so much that the data isn't out there somewhere, it's that you don't have the right combinations of data and the right volumes with the right mix to ask the kinds of questions we want to ask. So, in a perfect world, we'd have 1 billion tumors with 1 billion single cells and 100 regions per tumor and 100 time points. But given that resources are finite, really a lot of it is saying that we'd like to see more tumors with just a few single cells per tumor, but larger cohorts of tumor. We'd like to see more of the single cell data mixed with other omic technologies. I didn't mention it, but methylation data, for example, see more often single cell RNA-seq and DNA-seq together in the same tumor. It's really these combinations where I think we don't even necessarily know ourselves what the optimal study design is, but we know what's out there isn't it yet. And really that's where we think more can be done to really just ask that question: what should the field be doing to get the data that will best be powered to answer the kinds of questions we're trying to answer?
Great, thank you.
I was you talking about more single cell versus, more individual cells in one tumor versus more tumors. Do you see clear cancer stem cell signatures in your genomic analysis? Or is the population, if there are cancer stem cells, is the population of stem cells so small compared to the total number of cells that you don't see it's washed out?
I'd say probably the methods we're using, we're not seeing it so much, which doesn't mean it's not there. It may be that the kinds of approaches we're using, we are seeing more so the well-represented cell types, and the stem cells may be showing up in our phylogenies as something that's inferred as a past state, but that we're not actually seeing in the data. But again, I'm not saying it's not there, but at least the kinds of approaches we're seeing, it's not coming up so much for us.
Okay, well, I want to thank you very much for being willing to come today. And again, this is a lot of our people watch this asynchronously. And so I appreciate it. And it was very, very interesting to hear this perspective. And as you say, even if the core focus of the group isn't cancer, the methodologies are definitely very important and generalizable. And so I want to thank everybody for coming today. We'll look forward to maybe hearing more about your work in the future. And